So our first speaker this afternoon is a good friend of mine, Rosanna Peeling. Rosanna, I first knew her when she was with the National Chlamydia Laboratory, but lots of things happened since then. So Rosanna has been research coordinator and head of diagnostics research of the UNICEF, UNDP, World Bank, WHO Special Program on Research and Training in Tropical Diseases up until a few years ago, and then she moved to the London School of Hygiene and Tropical Medicine, where she started a program on international diagnostics as well. So Rosanna is really our pioneer of rapid STI testing, and it's my pleasure to welcome her.
Thank you very much, Michelle. And I first of all like to thank the Gairdner Award for inviting me here to give this talk, and also for to Grand Challenges Canada for funding my travel here. What I like to do in the next 30 minutes or so is just to talk about the development of tests for STDs, which of course preceded my time, but the idea that we need rapid tests for STIs actually started maybe in the late 80s, and then officially in the 1990s, there was something called the Std Diagnostics Initiative, and many of the founders are in the front row here. And then I want to just take a very quick look at the development. It's like 20 years in one slide, and then what's on the horizon. So this is the WHO stats on STIs. Sorry, it's going too fast. And we know that access to STIs is really limited in areas where there's the highest burden. And most of it is because there isn't a good laboratory infrastructure to do any diagnosis or nor screening. And so we are still at more than 1 million new cases a day in terms of the 4 curable STIs of chlamydia, gonorrhea, syphilis, and trichomonas. So I think many of the speakers this morning had talked about the need to improve case detection and finding. And so given that, when I was at WHO, at a meeting that King was at, we actually tried to talk with the funders about what is it that we really needed. And so there was USAID, Rockefeller Foundation, Gates Foundation, and the Wellcome Trust was there. And instead of always saying that we need something cheap and accurate, et cetera, I came up with this acronym, ASSURED, to stand for what we would like to have in terms of a benchmark for STIs. And of course, the words affordable, sensitive, and specific are all quite relative depending on what disease, what setting you're talking about. But since then, this acronym has even been used by donors. Now, when I talked to companies about what we wanted in terms of STI tests, a lot of companies would say to me, let me understand this. You want it to be cheap, you want it to be accurate, and you want it to be fast and simple? Well, let me tell you the truth. You can't have them all. You have to pick 2 out of 3. And so if we look at access and performance and cost, you could start with having tests that give you highly accurate results, but they can only be used in urban settings. And so they may be very accurate, but they're not cheap nor fast. Now, when you come down to something like an ELISA, then you could have something that is quite accurate, but cheaper and faster and simpler to operate. Now, if we want access to testing to be available to everyone, then we're only talking about these sort of very simple to use tests. But I think in making sure that they are cheap and they are simple and fast, you have to sacrifice some accuracy. So there's a trade-off that we have to think about. But nowadays, I think these tests are getting more sophisticated, and so now you could actually have this test, but you could insert it into a phone, and the phone would read the test, interpret it for you, and supposedly that is actually more accurate than the eye, and at least not as subjective as the eye, and you get a very accurate result, which then you can send that result to a central database, so you can be tracking the results of all these point-of-care tests. Now, how do we get there? As I mentioned, in 1990, the SDI was started and was supported mainly by USAID and funds from Rockefeller. And at that time, as King said, there was WHO was advocating syndromic management. So in a way, not very many companies decided to go into developing STI diagnostics because WHO is advocating that you treat all the causes of major syndromes with drugs and no need to use diagnostics. So in the end, it actually wasn't giving a very good message. But we realized that for chlamydia and gonorrhea at least, there were a lot of asymptomatic infections, and for syphilis as well. And in fact, you really need a diagnostic to find those cases, because without symptoms, people just won't come to the clinic. And so Rockefeller gave $1 million prize for a point-of-care gonorrhea and chlamydia duplex test. And that was in the early 90s, but in 1998, it was withdrawn with nobody having claimed the prize because the goalpost keeps moving, and they were demanding 95% sensitivity and 98% specificity. And so in the end, nobody got the prize. But that would have been quite an incentive for good development of good tests. So in 2000, I was asked to go to WHO at the end of 2000 to take over this initiative, the SDI, which at that time was, after being in PATH in Seattle, was in the Global Program for AIDS, and then was moved into WHO just a year before I arrived. And within 30 days, I was told to convene a meeting to look at what needs do we have for rapid tests. And so with that, we started to look at what is needed. And we did a survey that at the time, in 2000, there were 23 rapid syphilis tests, but 13 chlamydia and 12 gonorrhea tests. And so we produced a landscaping report together with some people from the Gates Foundation, and to advocate for what's needed. And at that time, we were already saying the landscape is very fragmented with very little funding and very little incentive, and not much is known about what is needed either in terms of technical specifications. And so we were hoping that with that, there'll be more advocacy around that. And the Gates Foundation then came to the fore, and in 2004 convened a global diagnostics forum, which went on for 2 years, and in 2006 it was published. The results from that forum was published as a special supplement of Nature, and that led to a whole lot of companies as well as funders also coming to fund point-of-care diagnostics, because with the forum, there were results that shown that even if you don't have the most accurate point-of-care tests for STIs, in fact, you could avert many, many infections as well as deaths with prenatal screening for syphilis. And so that was the kind of enabling environment that was happening across from 2000 and 2010. And in fact, King was our ringleader in all of these initiatives. And part of this initiative was at Johns Hopkins, and Tom Quinn is involved in that, and that's for chlamydia and gonorrhea tests. rapid tests. And at PATH, King was also leading a point-of-care test for global health. And so this is still ongoing. But in the meantime, what have we done? With the funds that we had from USAID and Rockefeller, we did some lab-based evaluation of all these tests. And then none of the chlamydia and gonorrhea tests really fulfilled the criteria for what we needed to go forward into field trials. So we only took rapid syphilis tests into field trials. But right after we finished that was the start with the Bush era, where a lot of the USAID money was withdrawn from STIs. And so we didn't have any more new money, but then the Gates Foundation came to our rescue and started to fund us for implementation research to see how we could put these syphilis tests that were good into the field, and how do we develop policy for adopting a point-of-care test. And so, and now in this, in the last few years, companies have developed a dual test that could, with 1 drop of blood, detect syphilis and HIV in the same kind of lateral flow format, which so is very easy to do, and hoping that this will be a very powerful tool for the dual elimination of mother-to-child transmission of HIV and syphilis. And we also have a combo treponema, non-treponema test as a rapid test. So lots of things are happening, but it's been a really, really long time and a lot of struggle. And Judy Wasserheit may recognize this quote, which is what Judy said to me at the end of this WHO meeting. She said, "I don't care how you do it, but just get this elephant to dance." And so I remember it through all my WHO days with all the trials and tribulations that we had. So this is, but with the Gates-funded 7-country project, we in fact, and King was also the head of our advisory committee as well, and also being involved in the Peru project again. And so we had in Peru, this is a picture taken with Patty Garcia just in March this year. And we checked out all the points that we've actually implemented syphilis testing, and they are still doing it 2 years after our project. So it was really great news, and a big tribute to Patty and her team. And here we have in Zambia, this is a toolkit that we developed out of this project, and our PI from the Elizabeth Glaser Pediatric AIDS Foundation is giving that to the First Lady of Zambia because she wanted to do some more about syphilis screening for pregnant women. And in Peru, we took the rapid test to the jungle and where we were doing screening right in the field. And in China, we had Premier Wen Jiabao actually announcing the incorporation of rapid syphilis tests into their national 10-year program. So the project was hugely successful, thanks to King and many of our advisors here. So I have to show this. This was in the national STI meeting, where all of a sudden we were asked to do, every province that came to this meeting was doing a special skit. And we didn't have some, but we thought that we could do a simple dance to a tune. I think I Could See Forever. I could see clearly now. And so I must say that I have to show this because King has given me a lot of really good advice over the years that had really led to the development of my career. And this is the single time that I was telling King, as well as Mike Cohen, Fred Sparling, and David Mavey wasn't paying any attention to me. I was hiding behind Sheila saying, "Guys, you need to use your red fans. We gave you these red fans." And we started to use our yellow fans, and I said, "Guys," so it was my advice to King after so many years. This is my only advice to King. Anyway, so that was the only time that I found King wasn't able to say anything back to me. Anyway, so one of the things that I also learned from King, which was also a result of the 2000 meeting, was King said, you really need to get into the internet. And I thought, at that time, we don't have social media, we don't have so many sort of web access, but he started this STI, disease literature reviews, which actually was very well subscribed. We had visitors from 111 countries that spoke 104 languages. And so for a long time, this had been very, very useful. And in fact, Hunter used to, I think Hunter agreed to be on the hotline for anybody who had problems, because my young secretary was receiving a lot of stories of sexual encounters leading to infections that she didn't know how to answer. So now what's on the horizon? Now, why don't we have good tests for chlamydia and gonorrhea? Now, part of it is actually, if we look at the fundamentals of having an ideal diagnostic, here we have, when you see the bug itself by microscopy, when you can detect DNA, you are very confident that the bug is there. Whereas if you have more indirect methods like antigen detection and indirect methods like serology, you are less confident because you're getting into cross reactions, etc. But ironically, these are so much easier to detect because there are many, many more antibodies in blood, and so it's very easy. You could measure it with very high sensitivity, and perhaps not as good specificity, but at least it's very easy to do. And it's you can detect it in minutes, whereas genome detection and even antigen detection takes longer. But for chlamydia and gonorrhea, we need to have antigen detection or DNA detection, and that takes way too long. You have to tell the patients to come back. Now, one of the other reasons why we don't have good tests, this is an evaluation that was done in China with these, the rapid tests, Clearview rapid tests, compared to PCR. And the results are that if you use a vaginal swab, you're only getting 30% of those who are truly infected. But it's not only that it's a test that you need to put the specimen on, but because you need to break up the chlamydia or the gonorrhea in order to get at the antigen or DNA, you need to do some pre-processing steps. So some of the rapid tests for chlamydia in the early days take about 7 steps before, and boiling, and then letting the specimens cool, and then filtering, etc. And it's too many steps before you could actually put it into the lateral flow strip, and then 15 minutes later you can read the results. So that's way too complicated for a rapid test and too long. But now, since I don't know whether this test by Cepheid is approved for use in Canada, but now we have a PCR platform, which is sample in, answer out. So you don't need very sophisticated technologies to be able to do this. And actually the results would come out in 1 hour and 45 minutes. And for TB, you can actually also read the rifampicin resistance as well. And this platform is available for MRSA, C. difficile, chlamydia and gonorrhea. That's already been approved and used in Europe. And then next year, they will come out with HIV viral load. So with this, you also have the ability to be able to have random access, which means that you don't have to finish a TB run before you could do a chlamydia run. You could actually just put them in as you need to. So this is great. And there are many things that are happening. This is for a rapid test for chlamydia, for HIV and syphilis. But when I asked the company to try and do this for chlamydia, they said it's way too difficult because of specimen processing and getting enough target molecules into these very narrow detection zones. But I think things are happening in terms of not only the technology, but governments being able to take more risks in terms of letting the test be outside of the laboratory. So this is a FDA approving the use of over-the-counter sales of HIV tests, and so now is available at Walmart. So you could, when you go shop. And this is just in. Yesterday there was a vote in the European Parliament, and they vote favorably for the home use of in vitro diagnostics. So this is going to be good for self-testing for HIV. So, and we were just recently funded by the UK Engineering and Physical Sciences Council to improve the early warning system for infectious diseases in the UK using nano-enabled technologies. And we're going to start with social media, looking at what's happening to try and detect early warnings of unusual fevers, etc., that would be before they come to the healthcare system. And the Gates Foundation and Grand Challenges Canada has been supporting a point-of-care diagnostics initiative in order to achieve the kind of flexibility that we wanted for the test to be available not only in lab systems but in villages. But what we wanted to have is a generic detection platform upon which different companies with different tests can make it work on that platform. So the closest analogy that we could use is that you could buy memory sticks from any company and it would fit into any computer. And so we want that kind of sort of flexibility so that the cost of development is much less if they could work on these systems. And so, and right here in Toronto, Warren Chan's been funded since this publication in 2007 on the convergence of quantum dot barcodes so that we could look at disease, not only the signature of pathogens, but their resistance hotspots that can be done. I mean, the power of the technology right now in terms of detection is phenomenal. So here I have a picture of something that can be hung over the ceiling of a big hall, for example, a shopping mall. And apparently you could tell with all these people in the shopping mall what sandwich each one is eating. So that's the kind of power that we're thinking of in terms of all these technologies. And each one of these nanodots, based on their sizes, would give you a different color. And with that, you could have many, many signatures that are encoded as almost like barcodes onto this small test chip, and you could read it on off your cell phone. And so things are really happening. You know, it's very exciting. But somebody said to me after I gave my last talk at the British Society for Parasitologists, you know, I talked about all these new technologies that will change how parasites are being detected. And the next speaker said, but the diagnostic landscape is a land of empty promises because there are so many publications of these fantastic technologies and we never see it on the ground. And funders realize that too. And why is that? It's because many of these innovations are from small and medium-sized companies, and they just don't have the ability to be able to then go into developing countries and seek out evaluation site, do clinical trials. They don't know how to get regulatory approval in over 190 countries in order to sell. And without that, they cannot even convince their investors to give them enough money to go through from this stage of development into the next stage, let alone having it be implemented in countries. And so the most urgent of these is that in 2011, UNAIDS and WHO actually pledged to put 15 million people on ART by 2015, and also to eliminate HIV infection in children by the year 2020. But as of last year, only 9 million HIV patients are on ART. So we have a big gap between that. And one of the strategies is to simplify CD4 testing. And so but we have all these point-of-care CD4 machines that have been developed over the last 7 or 8 years, and they're all stuck at market entry because these are all from small companies, with the exception of BD. And they need lots of help getting evaluated in clinical trials in different countries and getting regulatory approval. The same with viral load. We have a lot of small machines that could do viral load to monitor whether there's resistance to art, but also these machines are being, these companies are being stuck at market entry. So we've been funded by Untitled to look at how we could lower barriers to market entry and also to make clinical trials a lot more efficient, and then accelerate policy development and adoption, and also trying to come up with some quality assurance system for point-of-care tests, because once you deploy them into village level, it's very difficult to actually monitor quality to make sure that people are still getting the right results. And also in terms of regulatory approval of point-of-care tests, that's been a huge issue with some countries, including Canada, putting point-of-care tests as the highest level of risk and therefore requiring the most stringent in terms of evaluation and audits in order to get approval, in which case then approval would take far too long. So we've been tasked with the Gates Foundation and Grand Challenges Canada to look at how to harmonize and streamline regulatory approval in countries. And so far, in the not quite 2 years that we've had the grant, we've actually managed to get 12 Latin America countries committed to harmonization, 20 countries in Africa committed to harmonization, as well as 23 countries that are in the Asian Harmonization Working Party. So governments are committed, but there still needs to be a lot of technical work that's involved. And what we're trying to harmonize would be a single risk classification, a common registration file. So, in terms of right now, a CD4 test, every company that goes to a different country to apply for approval needs to fill out a different form. And so when you're a small company with only 20 people, filling out 190-some forms is very difficult, also in different languages. So we're trying to standardize that. Quality audits of manufacturing sites, how to produce the clinical evidence that's needed to ensure safety and effectiveness is very important, also post-marketing surveillance. So what we want to get to is a very streamlined path that is better, faster, safer, and cheaper. And we don't do this alone. We have lots of funders, and we're talking with lots of countries on how to do this. But I'm really grateful for Grand Challenges Canada for actually leading this initiative, as well as Untitled. And so hopefully we will be able to reduce that pathway from over 10 years, from once a product is ready, to less than 5 years. And so moving forward, I think that we have lots of really promising technologies. We still, I think, ideally would like to have better oral tests to screen for HIV and syphilis and maybe other STIs. For hepatitis C, we certainly have very good tests that could be used. We'd like to get a test menu for all these microchip type diagnostics, but we need to really define which pathogens we need to put onto these. We need to define better the needs and to be able to create an enabling environment to accelerate market entry and patient access. And also, we really need concerted action to know how to strategically place these new point-of-care technologies to maximize their impact. So that's where we are now, and thank you, King, for all your leadership. Thank you.